You don't need a nutritionist to tell you how bad processed food is for you. And I'm sure you've heard the star of the processed food industry, high fructose corn syrup. We, as one of the smartest species on earth, have managed to take fructose, a common monosaccharide found in all fruits, and made it into a major mediator of non-alcoholic fatty liver disease. But what if we told you that before us, Roman geese were also subject to steatosis due to overconsumption of fructose? How exactly did fructose trickle down from Roman geese to liver diseases in the developed world?
A Roman chef from the 1st century AD, Apicius, was a member of the Roman elite at the time, whose full-time job was to make fancy recipes. While making a dish called foie gras, which translates to fatty liver, he discovered that overfeeding geese with dried figs resulted in them developing fatty livers, which worked brilliantly for the dish, but not so much for those who ate it. But why did they use figs in the first place? Figs have high fructose, which is a monomer of sucrose and is found in almost all processed foods today in the form of high fructose corn syrup. High fructose corn syrup usually gets a bad rep, but why? Fructose is essentially a form of sugar which, when metabolized, causes a spike in blood sugar levels. But repeated consumption results in insulin resistance, weight gain, and you guessed it, obesity. Similar to the geese, it can also cause fatty liver disease in humans.
Non-alcoholic fatty liver disease is a range of liver diseases that is characterized by excessive storage of fat in the liver cells. While fatty liver diseases are usually associated with overconsumption of alcohol in a short amount of time, non-alcoholic fatty liver diseases, as the name suggests, happen to people who drink little to no alcohol. In fact, Apicius' genius recipe led people to believe that the fructose content in figs directly led to the development of fatty liver in geese, which is where they went wrong.
Fructose does not directly cause fatty liver, but it triggers a chain of metabolic reactions that may end with non-alcoholic fatty liver disease. While we thought that sucrose, glucose, fructose, and consequently high fructose corn syrup are all metabolized the same, that is not the case. Glucose is processed in the body differently. The consumption of glucose results in a rise in blood sugar, causing insulin levels to increase in response. This insulin helps blood sugar enter the cells in the body to be used for energy. Any excess glucose is stored as glycogen through the liver-mediated process of glycogenesis, or with the help of insulin converted into fatty acids circulated to other parts of the body and stored as fat in adipose tissues.
So why is fructose different? While fructose doesn't directly cause fat accumulation in the liver, the enzymes that work to metabolize it is what acts as a mediator in non-alcoholic fatty liver diseases. Fructose is first phosphorylated into fructose 1-phosphate, a job done by the fructokinase enzyme. This is followed by the metabolism by another enzyme called aldolase B to generate D-glyceraldehyde and dihydroxyacetone phosphate. From this stage on, fructose metabolism is similar to glucose metabolism and results in the generation of glucose, glycogen, and triglycerides.
Fructokinase comes in different isoforms, but the main isoform in the liver is fructokinase C, which phosphorylates fructose rapidly without any negative feedback control that leads to a drop of ATP and intracellular phosphate. The fall in intracellular phosphate activates adenosine monophosphate deaminase, an enzyme that converts AMP to inosine monophosphate, resulting in purine nucleotide turnover that culminates in the formation of uric acid. The fall in intracellular phosphate during fructose metabolism causes a transient block in protein synthesis, induces oxidative stress, and mitochondrial dysfunction, all of which have a key role in mediating fat accumulation in the liver.
Fructokinase A is the second isoform of fructokinase and is more ubiquitously expressed, but it differs from fructokinase C in that it phosphorylates fructose less efficiently and does not cause significant ATP depletion. In a study, experimenters blocked fructokinase A in mice to see how the body reacted, as compared to mice who had both types of fructokinase. It was observed that mice lacking both fructokinase C and A were protected from fructose-induced fatty liver, while fructokinase A knockout mice developed worse fatty liver compared to wild-type animals despite consuming similar amounts of fructose. These animals also show evidence for greater metabolism of fructose through the fructokinase C pathway and have higher uric acid levels in the liver. These effects increase the lipogenic enzymes that disrupt the tight junctions in the intestine, making the gut more permeable to endotoxins or lipopolysaccharides that are known to trigger fatty liver formation.
Now imagine a world where anything and everything you eat has high fructose corn syrup, or some form of excessive fructose in it. While this would open up more job opportunities for the fructokinase C enzymes in your gut, it will also subsequently increase uric acid levels in the body and make the gut more permeable to endotoxins that trigger fatty acid formation. But do we really need to imagine? In highly developed and developing countries where most processed foods and beverages rely on fructose as a sweetener, non-alcoholic fatty liver disease is the most common form of chronic liver disease, affecting about 1/4 of the population.
The rise of availability and affordability of processed food, coupled with the hypnotic effect of globalization, has brought about a culture that has convinced us that eating real food is healthy when it should be the norm. For people who can afford to choose what they can put into their bodies, it comes down to this. In the world where you're surrounded by dried figs, do you really want to be the next serving of foie gras?